Well, good day to everyone joining us and welcome to today's XTalks webinar. Today's talk is entitled Patient-Centricity in Neurodegenerative Diseases. My name is Ryan Muse and I'll be your XTalks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Fortrea, who developed the content and brought everyone together today. Fortrea is a leading global provider of clinical development and patient access solutions to the life sciences industry. They partner with emerging and large biopharmaceutical, medical device, and diagnostic companies to drive healthcare innovation that accelerates life-changing therapies to patients in need. But now, without further ado, I'd like to hand the mic over to our moderator for today's event, Vikas Mohan Sharma, MD, Global Head of Neuroscience and Ophthalmology at Fortrea. Vikas, you may begin when you're ready.
Thank you, Ryan. Good morning, good afternoon, and good evening to people all around the globe. With great pleasure, I must talk about this webinar where we got together to talk about a topic which is very close to not only my heart, but I think all the speakers here. And that is, how do we make sure, using neurodegenerative disorder as an example, that we do not forget the most important voice when we are trying to create new treatment, new modalities, new solution, and that is the voice of patients and their near and dear ones. So the topic for today's webinar is patient-centricity in neurodegenerative disorder, but what we're going to talk about could be applied simply to any clinical disorder or any conditions in CNS or even beyond that. And I'm so grateful that to provoke this discussion, we have a great set of speakers today. Myself, I'm a clinician, but I've learned a lot in my journey as I've understood what truly is meaningful to patients versus what not necessarily driven by science. And in my current role as the Global Head of Neuroscience and Ophthalmology at Fortrea, that something keeps me grounded all the time. To drive the discussion, we have three speakers, starting with Dr. Bill Potter, which anyone in the industry needs no introduction for. He has been in NIMH for a long time, has been industry, led early clinical development programs and departments in large pharma, has been consultant in the industry, and most importantly, has been a true and trusted partner, is making sure that other stakeholders understand that science must be meaningful. He's going to challenge right off the bat about what happens in industry vis-a-vis taking the decisions from drug discovery to development, how we remain patient-centric. Then we go to Dr. Hartmut Kohl, who is another force in the field, and he has been in industry long. He's been trained chemist. He has, besides making sure that he waves the flag for precision medicine, what he'll talk about, he also co-chairs the steering committee for the biomarker consortium at FNIH and many other similar collaborators. Hartmut will use the approach of precision medicine, specifically in CNS, to put a spotlight, how to remain patient-centric there. And then finally, last but certainly the most powerful voice would be Shana, who would be representing, Dr. Dodge, would be representing the Association for Frontotemporal Degeneration, and she's the Director of Research Engagement, and she would help us bring it all together before we open for discussion. Now, we are in a virtual setting. I understand that the technology doesn't allow for all of us to be in the same room, but let it be a dialogue. Don't hesitate, as we go along, to put your questions and even comments, what comes to your mind, in the chat, and hopefully that would be a virtual interactive discussion. At the end, we keep some time to take some questions. So with this, without further ado, I hand it over to our first speaker, Dr. Bill Potter.
Thank you very much, Vikas, and it's a pleasure to be here. Just very briefly, I continue to be very involved in serving as a consultant and even a drug developer myself. So the history of my background, I think, will help put into perspective why I believe it is so important to think about what, from the patient view and the patient's family view, is most important when we invest in novel treatments. Okay, this is a huge decision. Of course, we all understand medical need is important, but who defines medical need? Obviously, a lot of that comes from patients and their families. Then there's the scientific basis, which Dr. Kolb will speak much more about, and that is the rationale for what we call a target, a biologic target. And one of the reasons for focusing on neurodegenerative diseases is because they include, particularly when there's a genetic component, a scientific basis for understanding the pathology. This is a complex issue, and I'll return to it so you can understand the difference between disease-altering treatments and symptomatic treatments. And this is an important distinction. Then, of course, there's the economic path. And it's true that, at least in the United States, we have the rare disease program, which helps make it, shall we say, somewhat less expensive and more efficient to develop a new treatment. But frankly, the economic path and the rationales are not always strong enough to go after a particular medical need, okay? And that's where the advocacy from patients and disease focus groups becomes so important, both at the level of raising awareness of the need for the treatment, arguing do you really just go for a cure, or do you push for the idea that even if there's no cure, there are symptomatic interventions that will greatly improve the quality of the life? And all of the following talks will touch on aspects of these themes. I just briefly want to use the Alzheimer's experience on this rather complex slide to highlight a couple of points. And because neurodegenerative diseases like Alzheimer's are progressive, okay? And so therefore, you have early phases where we don't even see any evidence of disease. Obviously, if we could, and we knew before anything happened that we could stop a disease from ever emerging, we would do it. But right now, that is something in the future. So where we now enter in the real world in terms of Alzheimer's treatment is in the earlier stages where we call it pre-dementia. But so think of any of your diseases. Maybe there's some suggestion that something is wrong. Obviously, dementia means your cognition, your memory isn't working as well. But, and so we have these milder symptoms. And ideally, we would say, well, even at this stage, we could intervene with the treatment and stop the progression. In other words, you might have them, but you won't get any worse. So that idea of slowing the progress, shown at the bottom of the slide here, would be targeting the primary underlying pathology of the disease, okay? And Dr. Kolb will talk about some of this. But at the same point, symptom relief remains very important, a theme to which I will return. And here is where particularly the patient and family voice is critical. I'm not going to talk about the later phase because I think we are always most excited about either slowing the process by hitting the underlying disease process or going after the symptoms and finding ways to relieve the symptoms. So the points at which input from patients and families can be most important are at the basis, at the very beginning, selecting the target. Because at this selection, you might have a target, like in the field of cancer, where the treatment can be very risky. And so you have to trade off if you want to hit a particular biologic target, whether the risk is worth it. And the worth of the risk really is in it is in the purview of patients and family. Because who decides on risk benefit? You, the people. Okay. Dr. Cole will go greatly into the middle part of the development after targets are selected and what we need to understand and study a target. We call them critical biomarker developments, and I'll show this briefly on a slide where this occurs. And that's very important, and that involves the participation of patients even outside of getting treatments. So it's a critical point for input. And then finally, the most obvious area of importance is where we are focusing on which symptoms and how to best measure them. And we do this in drug development in something called phase 2. So I'm going to show you briefly in this complicated slide the whole development process from beginning, target ID, which I talked about, all the way to the right to a drug coming on market. So what I wanted to emphasize is that these phases, when you think about what biologic target am I going to try to influence, to even getting into the first human phase, which is called phase 1, here in the middle of the slide, on average takes 6 years. And if you were lucky, we can get a drug to market 6 years later, the way we work. But in reality, like the drugs in Alzheimer's, it's taken 20 years to get one that was good enough to get to market. Okay. And the other important complex thing, which has been recently included in drug development, which wasn't true 20 years ago, is that we want to have a biomarker of drug effect in the brain. And then I'm sorry about the phone in the background. I hope it's not. We need a parallel development process in people to find these biomarkers. And Dr. Cole will be talking about this in great detail in the next talk. So I'm going to give you 2 quick examples, just I hope to make it clear how even in programs that we're now thinking about, there's a lot of room for patient input to make this not theoretical, but practical. So the first one I'm going to talk about briefly is what's called apathy in Alzheimer's and other neurodegenerative diseases. And the second would be something called negative symptoms in anhedonia and schizophrenia, just to give you an example. And many people view schizophrenia as a neurodegenerative disease. Okay. In the first case, we are interested in what's called apathy, but the case has been made by clinical experts and investigators, not patients and their families. And at this stage, we rely on efficacy scales that, frankly, were developed for other purposes. So very briefly, the background is a lot of Alzheimer's patients, 70% of them actually probably have what we call apathy. And what it really is just not being interested in things, not being motivated to act. And so I'm clicking down to just the lower motivation. They don't want to do things, no emotions are expressed. And there's the medical case is the association between apathy is with accelerated cognitive decline, demoralization of caregivers, and hastening of institutionalization. However, even though we have diagnostic criteria, we have a big open question on how best to measure. And you can imagine something as subjective as apathy could well benefit from input from families and their patients. Okay, that's just a quick example. And I'll give you one other very quick one. And that is the area of something in this way related to apathy, they're called negative symptoms. That's just being withdrawn as opposed to what usually people think of with psychotic disorders where people are wild and irrational, or anhedonia. But it's very similar in a sense to the concept of apathy. And again, not to go too much into detail, on the left in blue, there are all these scales, and each one tests as I'm showing, some slightly different concept. But we have no way of knowing at this point whether these really are the best scales. And since we are still using scales that were, most of which evolved 40 or 50 years ago, there's a huge opportunity to perhaps generate better ways of measuring what we believe might be affected in a positive way with new treatments. So I hope these examples make clear that there are real opportunities, meaningful opportunities for patient input. And on this last slide, I just wanted to emphasize that as a whole field, as part of this universe of what it takes to do drug development, obviously we have academia, we have industry, we have government funding, and all to generate in the middle all the standardized tools to do this best. But what we've now appreciated in the last several years, using the examples I've shown you, is how important the input from patients, their families, and foundations is to selecting the target, making the best case for it, and figuring out the best way to study this in the people who are the ultimate recipients of any treatment that is developed. So thank you for your attention. I'll stop at that point.
Thank you so much, Will, for setting the stage so nicely. I mean, you raise a couple of points which I think the crux of the matter. You straight off the bat start by talking about what truly is meaningful. And that's a question which I often, in a provocative but I think sometimes blunt manner, put at the medicine. That did modern medicine begin well? Because while it helped across the last centuries, billions of patients and their caregivers, it remained physician-driven, top-down, centric, which exactly is what we're challenging at this moment. So this becomes absolutely important that we talk about the meaningfulness and also acknowledge that while we in industry invest a lot in robust science, but robust science by default is not meaningful. And that's something which we'll talk about. This is a great opportunity for me to thank you and now invite next speaker, Dr. Hartmut Kohl, who's going to take it from here to go a little bit more into trenches and talk about how do we remain robust with science, but remain patient-centric when we are precise in our strategy in developing right tools, right biomarkers for taking the right drug to right patients. Over to you, Hartmut.
Thank you. And I want to start by thanking everybody who's joining this webinar and for your interest in this topic. This has been driving us here on this call for many decades now. And so I want to focus on precision medicine, and which at its very core is patient-centric. And there's no better place to start than with how the FDA, the Food and Drug Administration, defines precision medicine. It starts by pointing out that most medical treatments are designed for the average patient as a one-size-fits-all approach. And that's certainly true in neuropsychiatry at this point still. In precision medicine, really, is a paradigm shift in the way it switches the focus on the patient by matching the right treatment to the right patients based on the biomarkers or genetics or real-world data, digital data, and other data. So basically matching the treatment to the patient, that is the core of precision medicine. It seems obvious, and this also is being done in oncology now more and more, but it's not as obvious and easy to do in neuroscience because the brain is pretty complex. So as recently as like 10 or 11 years ago, many these disease-modifying therapies against Alzheimer's disease, for example, anti-amyloid therapies, they were enrolling patients based on clinical symptoms. And it turns out that about 15 to 30% of these patients in those clinical trials that were enrolled based on clinical symptoms were amyloid negative. So they didn't even have the target that was being targeted. And that's why many of these early trials failed. This has changed over the last 10 years. And we now have three FDA-approved drugs and approved drugs that target amyloid and that are based on the realization that amyloid needs to be present in these patients. Right? And this was really driven by the groundbreaking work of the Biogen team, headed by Samantha Bhatt-Haberlein, and who used this approach and to match patients that are amyloid positive to anti-amyloid therapy, right? And then the FDA recognized that amyloid is a reasonably likely surrogate endpoint, meaning that if you remove amyloid from the brain, you're reasonably likely to have a clinically meaningful impact. So this realization of the importance of the pathology as measured by biomarker measurements is what led to this sea change, right, in treatments, starting with aducanumab, and now, of course, having led to lecanemab by Eisai and donanemab as late as last week. We got approval for Kisunla. The FDA, as you can see in the label, states that you should confirm the presence of amyloid pathology in these people. And for donanemab, it goes one step further even, saying that you should consider stopping dosing based on reduction of amyloid. Right? So clearly, biomarkers and therefore patient centricity is the key here to these novel treatments that actually are disease modifying in Alzheimer's disease. So the question is, how do we get there? And it really is based on the work of an army of researchers over the last decades, 2 or 3 decades, have looked at first the pathology in Alzheimer's disease in autopsy samples, amyloid, and then here shown here tau. This was done by Bragg and Bragg, a husband and wife research team that looked at the spreading of tau pathology, NFTs here, as the disease progresses, using, of course, samples from different patients. And this then was further supported using in vivo. This is autopsy, of course, is postmortem. We can also do the same thing in vivo through imaging, PET imaging in this case, where we have had now amyloid imaging agents for the last 2 decades, and starting with Pittsburgh B, Carbon-11 Pittsburgh B, and others, and also as of 10 years ago, also tau PET, starting with flutalsapir that my team and I developed at Siemens. And you can see that amyloid and tau can be measured in individual patients. Each slide here is a patient, but in a living patient that we could image and determine, and we can look at the progression of the disease in these patients. And that led to the famous Clifford-Jack graph that shows the changes of biomarker patterns over time. And you can see that it really begins with amyloid, which can start decades before cognitive symptoms occur, and then tau pathology on the right side is more in line with the actual severity of cognitive symptoms. And in between the amyloid pathology and the tau pathology, we can measure fluid biomarkers, phospho-tau fragments, A-beta, and neurofilament light, and other GFAP and other biomarkers that we can measure there. Right, so amyloid and tau are key, and they can be measured on an individual basis in patients. And this had then led to a biomarker framework, which is system agnostic, but it defines the disease of Alzheimer's disease through biomarkers. Right? And the biomarkers focusing primarily around amyloid and tau. These are the core biomarkers. And then also recognizing that other changes happen too, like neurodegeneration or inflammation, and also co-pathologies like vascular injury or alpha-synuclein, not mentioned here, also TDP-43 and things like that. Right? So we have defined the disease now through biomarkers that can be measured in plasma or CSF and through imaging. And probably the most common tools, techniques are now amyloid PET, tau PET, A-beta in plasma and CSF, and also phospho-tau fragments such as p-tau181 and p-tau217. So these things we can measure in individual patients that allows us then to diagnose and stage a patient, and therefore match this patient with the treatment. And I should also, let's back up a little bit and see how amyloid plays a role in Alzheimer's disease. And so here we know now that with aging, amyloid rises. So if you are 80 years old, in all likelihood you have amyloid in your brain, even if you're cognitive normal. If you have amyloid, if you have slight symptoms, then in all likelihood that is linked to amyloid. And if you are 90 years old, you have a 50-50 chance of having amyloid in the brain. So amyloid is increased with age, and amyloid is prognostic for decline. But if you have a lot of amyloid, you go down quicker than if you have no amyloid. So amyloid is a prognostic biomarker for decline, and we can measure that now in living people, and therefore determine that. Tau, on the other hand, it predicts decline. So if you have tau, it's bad, and and but also it tracks decline. As you can see here, if you are amyloid positive and tau positive, shown here by this green line, you go down pretty fast. We can see how quickly over a period of 2 or 3 years you cognitive symptoms become worse. And but tau also tracks the decline. Right? So tau PET is a tool for predicting decline and tracking it, and therefore it could also be a surrogate endpoint, which we are currently working on as well. So amyloid PET has been around, as I said earlier, for 20 years. Beginning with Pittsburgh B, which is not convenient because it has a half-life of 20 minutes. So there's a new version of it now, which is was developed by AstraZeneca, and it's called NAV 4694, and that is an F18 version, structure shown here, very similar in structure, which allows you to measure amyloid in individual people's brains and therefore to diagnose them. You can see in healthy volunteers, you have less amyloid than you have in AD subjects. Right? And so that's can be tracked. How does amyloid now relate to tau? And as you can see, if you have little amyloid, you also have no tau. Right? So they go hand in hand. If amyloid goes up, tau goes up too, but delayed. Right? And you can see that here in this chart, where you on the x-axis you plot the centroid levels of amyloid, and on the y-axis the percentage of tau positivity. And you can see that nothing much happens up to like low amyloid levels of 30 or so centroid, and then after 40 or higher, you shoot up, right? And in other words, tau pathology becomes more and more prevalent. And so if you have a high level amount of amyloid in the brain, you also are incredibly likely to have tau in the brain, right? And this is what Keith Johnson, professor at MGH, calls cartography, because it really shows how amyloid, in a way, induces or is linked to the production of tau fibrillary tangles, which are detrimental to the brain. And not just tau goes up, but also other biomarkers, as shown here, phospho-tau fragments, GFAP, and which is linked to inflammation and other biomarkers. I don't have time to go into all this detail, but we understand the pathology and the development of pathology longitudinally in Alzheimer's disease very well, and that's been helping us in the development of these therapies that exist now. The FDA has been a very knowledgeable and important partner in this, and they have very early on defined the biomarker qualification program, so tools for classifying biomarkers, and that's been really a guidance for all of us, right? So they define biomarkers that measure the disease presence and status, but also measure aspects of response to treatment. And so you can look here at diagnostic markers, prognostic markers, already mentioned them, predictive markers, surrogacy markers. And in AD, we have it really covered, right? Because we have, with A, T, and N, we have tools that allow us to measure these aspects on an individual patient-centric level, right, for each patient. And so the FDA is very aware of this, and they have, in a way, catalyze this, and that's where they have used amyloid as a reasonably likely surrogate endpoint, right, which then led to the approval of aducanumab, which led to this whole change in the landscape. Jeff Cummins last year summarized an overview of the various drugs that are being developed in AD, and you can see as of last year there were 141 unique drugs in phase 1, phase 2, or phase 3 studies, and the precision medicine tools that many of these programs use are amyloid as an inclusion criteria and amyloid tau PET as a staging and tracking biomarker. Roger Nitsch at CTAD also analyzed what sets apart the anti-amyloid trials that were positive, so that had a successful change or slowing of the dementia scores over time, whereas negative trials that did not impact the dementia scores over time in a statistically significant fashion. And the key differentiation between the positive trials, so for aducanumab or for lecanemab or donanemab, versus the negative trials, is the tremendous lowering of amyloid in these people. Right. So if you go, so these positive trials, they are characterized by ending up at 25 or less centiloids in the people. Right. So in other words, clearly amyloid is a key element in the Alzheimer's therapy, but also in the progression of the disease. And if you reduce it to very low levels, you have a higher chance of having a positive impact than if you don't achieve this goal. And that's what this data here shows. And now moving quickly to fluid biomarkers, because we are in the lucky position where we can use plasma assays now to get an estimate of if there's Alzheimer's pathology present or not in patients. And my team, but also the little team, have developed a plasma P217 assay that allows us, that is associated with amyloid pathology. And it's basically accepted now that A beta negative people have lower levels of P217 in the blood than A beta positive people, as shown here. Right? So if you have a low level of P217 in your blood, you're in all likelihood amyloid negative, so you're fine. If you have a very high level, you're in all likelihood positive, and you have to then look into treatments. And that's shown here. So you can see here, we have now defined two thresholds, a higher one and a lower one. If you're above the higher threshold, you are in all likelihood with 90% or higher likelihood positive for amyloid. If you're below the lower threshold, you're in all likelihood negative. And people that are in between, the yellow ones here, they will then go on, so they're in the gray zone, they will go on to an amyloid PET scan, which will be definitive, so you get an idea. Right, so we can now establish a workflow whereby we take a blood test in people, we determine whether they are positive or negative, or gray zone. If they're in the gray zone, they get an amyloid PET scan. So we can basically determine whether a given patient is positive or negative, and in a very efficient way. And that's being done in a very innovative way in the AHEAD study run by Risa Sperling, Keith Johnson, Paul Layson, and others that looks in the plasma assays for patient enrichment and then A beta PET to confirm the presence of amyloid in the people. These are people that are cognitively unimpaired, so normal, healthy people, and but that are A beta positive. And then they're using amyloid PET, NAV4694, and tau PET, MK6040, to track the disease. They have two arms, the A3 arm that looks into lower amyloid, like 20 to 40 centiloids, and then the more elevated amyloid arm, A45, that looks into cognitively normal people with more amyloid in the brain. But and the treatment is with lecanemab, and the goal is to show that lecanemab slows progression of and prevents cognitive decline in these subjects. That's running right now, and we hopefully learn more about this soon. I'm almost done with my talk, so I just want to highlight that it's not all about amyloid and tau. There's also other pathologies that we need to worry about, such as TDP-43, right? That is present in a lot of brains, alpha-synuclein, neuroinflammation, and so on. And we now have biomarkers, at least leads for biomarkers for imaging, and also fluid markers that tackle these things. So in all likelihood, we will be able to have a similar impact in other diseases such as FTD, and which Dr. Deutsch will talk about, and PD that will deal with these biomarkers. And the last slide, just to show where we headed in patient-centric precision medicine in neuroscience. In AD, I think we are in a good place. We are able to diagnose, stage, and track a given patient and match this patient to therapy. In other diseases, we are not as good yet, but we're getting there. Like in PD, alpha-synuclein plays a role, and we can measure that now using synuclein aggregation assays in CSF, and hopefully soon also PET. Then we also will have more precision medicine trials that are driven through blood tests and through imaging and AI supported data analysis tools. And that is happening in AD already, and is also happening in other diseases, even in neuropsychiatry, where we're looking into complement inflammatory markers, BDNF, and neurotransmitter dysregulation. We also, and there, thanks to the FDA, we have alternative pathways, accelerated pathways to registration. FDA, again, I mentioned earlier, has accepted amyloid PET as a reasonably likely surrogate endpoint, and we are currently working with the Critical Path Institute, or the CPET initiative, Diane Stevenson, and to establish tau PET as a surrogate endpoint as well, and in collaboration with the FDA. And then drug development is shifting to a more biomarker-driven approach. I explained this for AD, and this will also be happening in schizophrenia and others. And then diagnostics will become common, right? And might even be rolled out to your primary care provider, where you take a blood test, and then you get an AD score that then links you up to treatment, similar to what's already happening in cancer, right? If you say have elevated PSA, you do a biopsy to see whether you have prostate cancer. So these things will happen and become more and more common in daily medicine. And this is the end of my presentation. Thank you.
Thank you so much, Hartmut. Your presentation really raised some very important points, starting from defining what is the precision piece in precision medicine. And using biomarker development as an example, to me, you raised the fundamental question that how do we help our patients, specifically in front of in neurodegenerative disorder, help patient identify and ask this question: how early can I know that there is a window of opportunity to intervene? And that requires massive investment from industry and academia and everyone. And Alzheimer's took more than 2 decades to do so. And hopefully what we learn from that will make it faster in future. You also raised a very important point about that is the scalability and accessibility of biomarkers. For what used to be CSF-based, fluid-based, to hopefully in future blood-based and maybe one day digital surrogates of biomarker, imaging came in front to the rescue. And while the scalability and accessibility is still to be worked upon, is a far better tool than what used to be only biofluid-based tools. Great opportunity for us to now invite our final speaker, Dr. Shannon Lodge, to educate us, and I really mean educate us, people in industry and everyone. How do we listen harder, and how do we involve at each stage the voice of the patients and caregiver so that we remain scientifically robust but meaningful?
Thank you so much, and I'm just shifting some things around on my screen so that I can see this appropriately. Thank you so much for having me, and it's such an honor to sit on this panel with such esteemed colleagues, and I will try to be mindful of my time. I am here from the Association for Frontotemporal Degeneration, and just a few words about who we are at AFTD. Our mission is to improve the quality of life of people affected by FTD and drive research to a cure, and we have 5 mission pillars, which include research, awareness, support, education, and advocacy. If you're not familiar with FTD, it's an umbrella term for a group of related disorders that are relatively rare, progressive, and fatal. There are heterogeneous symptoms that include changes in behavior, language, and motor symptoms. And although we currently don't have any approved disease-modifying or symptomatic treatments, there's been substantial growth in clinical trial opportunities and subsequent interest from stakeholders in understanding the patient or family voice. So, engaging with persons diagnosed and their care partners in clinical research has both practical and ethical benefits. And today I'm just going to talk a little bit about the practical or logistic benefits, but I hope it goes without saying that patient centricity is also the ethical and humane way to approach research. In my role at AFTD as Director of Research Engagement, I'm privileged to work with the best and brightest scientists in neurology who are developing exciting and innovative interventions for this devastating disorder. But the science itself can only get us so far. And I'm going to go through just some examples of significant insights that people with direct lived experience of FTD have had that have had huge implications for the FTD drug development process. And I'm going to start with one example regarding genetic testing. Many of the current drug trials for FTD are for those with FTD that's caused by a genetic variant. So that's a subset of an already rare disease. And these trials are not easy to recruit for. AFTD partnered with the FTD Disorders Registry on the largest community survey of people with FTD, their care partners and family members, and I'll be presenting some data throughout my slides from this FTD Insight survey. And one thing this survey revealed was very low genetic testing rates. For example, of those folks diagnosed or those who have a close biological relative with FTD, only 16% have undergone genetic testing. And the pie chart on the right shows that of those, a significant number, about 17%, aren't sure what gene they carry, those that believe they carry a gene for FTD. So this has obvious implications for people even knowing whether they're eligible for a clinical trial. This also has helped us inform some of the sponsored genetic testing programs that sponsors have stood up to ensure that they have features such as both pre and post genetic counseling, so that when people receive those results, they can actually understand them and take action for things like potentially enrolling in the clinical trial. Another point that I wanted to make is something related to what Dr. Potter was speaking about. I was at a scientific conference last year, and really brilliant scientists were discussing how to best measure the ability to stop or slow the progression of FTD using different statistical modeling. And a person who has lost family members to FTD and is also at risk of being FTD themselves came up to the microphone and simply said, "Hold on, we don't want to stop the progression once quality of life is a low point." And I've heard from other people, some of whom don't want to enroll their loved one in a clinical trial specifically because they don't want their loved one's progression frozen at a certain time period. And again, this has implications for things not only including recruitment language, but what is the right time to intervene? Another set of concerns that have arisen based on input from family members is the issue of anosognosia, or lack of insight. So patients and families impacted by FTD have highlighted the fact that what's clinically meaningful is different depending on who you ask. And this is a really busy slide that just shows what symptoms the respondent finds most distressing, with care partners on the left and persons diagnosed as respondents on the right. And you see significant differences. So some of this may be related to different perspectives. Persons diagnosed may simply be distressed by different symptoms than what their care partners are distressed by. It may also be an artifact of anosognosia, the fact that persons diagnosed may not have insight as to the impact of some of their symptoms. And again, this has significant implications when it comes to selecting clinical endpoints. When we talk about what's most distressing or what's clinically meaningful, who are we talking about? Are we talking about the care partners? Are we talking about the persons diagnosed? Another set of concerns comes to barriers related to clinical trial participation themselves. While many sponsors generously cover the cost of travel trial sites, especially in a rare disease like FTD, where those clinical trial sites may be few and far between, is a significant burden for people with FTD and their loved ones, some of whom have difficulty simply getting in the car or going to a local doctor's appointment. And here are some insights that those with FTD have shared. The fact that they would be more willing to participate in a clinical trial, so these are responses from folks that said, "I'm not willing to participate," if a professional could come to their home for some of those aspects, things like blood draws, or if they could complete some interviews or assessments from home using a computer or smartphones. Some specific quotes from people with lived experience included, he would have participated if he could have participated and not have to travel so far. The fact that participating is not physically possible, or that we're too far into this disease. So I would encourage sponsors to think beyond simply the money side of supporting travel, and what other concerns might people have, and where might we be able to decentralize some of that participation. Another set of concerns, and something that only people with direct lived experience can really elucidate, is the impact of symptoms themselves. And here's a case of how that might impact things that are involved in participating in clinical trials. The left hand or teal bars are what people diagnosed have reported in terms of the impact of symptoms, and the gray or right bars are what care partners have reported. And you can see really the impact across the board and on a lot of domains that are impacted when it comes to participating in a clinical trial. So things like being able to manage appointments, managing medications, driving or using mass transit, physical activity, or interacting with new people. And you can see how these might all have implications for clinical trial participation. I wanted to highlight the fact that people with lived experience can tell us what words resonate and what words offend. And that really has a lot of implications on clinical trial participation. One thing that we really encourage people with FTD and their loved ones to consider is brain donation. And we encourage them to discuss it early with their family members so that everyone's needs and desires can be discussed. And we also believe that brain donation is one of the key scientific participation aspects that people can that may or may not be able to participate in clinical trial can explore. And I had one woman speak to me and say, you know, I took your advice. My husband and I discussed brain donation early on in his disease. I've received the packet of information about our family considering this. And the first question on the packet is name of deceased. Well, my husband's not deceased yet. And that was so upsetting that she threw out that packet of information and they won't be pursuing brain donation. That small word, name of deceased, really turned them off from that that participation. And I've heard similar words used in consent forms can really get in the way of people participating. And mindful of time, I just wanted to briefly talk about some specific concrete ways that our patient advocacy organization partners with industry, with academia, with CROs to amplify the lived experience and to promote patient centricity in the drug development process. One is that we held, in partnership with the FDA, a patient-focused drug development meeting. The recording of that meeting and the voice of the patient report live on our website and on the FDA website. And it's a great way to really dive in and see testimonials and understand the lived experience of FTD, for anyone who's interested in pursuing an FTD program. We're also blessed to have a patient registry that has a wonderful qualitative and quantitative data on the lived experience of FTD that we're always happy to share with industry partners and find ways for them to leverage the registry to gather data that they're interested in terms of lived experience. AFTD also helps find folks that can participate in company-led focus groups or advisory boards. We have also found people that can speak at company staff meetings, at lunch and learns, at company retreats. And we've gotten really good feedback from industry partners in saying that it's really helpful for their scientists to hear from people with direct lived experience. And it really helps remind them why they got into working in this neurodegenerative field to begin with. We, along with a lot of other patient advocacy groups, also host our own advisory panel made up of people with direct lived experience that industry can sponsor and help contribute to the discussion guide for. We work to bring people with lived experience to scientific meetings to ensure, like the example I said about slowing progression, that people with lived experience are contributing to that scientific discussion and are able to see what's happening at the forefront of the science of FTD. We're always looking for ways that industry can informally engage with our community. So, for example, we hold an annual education conference that is community-facing, and we look for ways for industry partners to be in the room and to, speak with community members by the water cooler. We also offer preclinical touch points. So even for companies that are considering going into the FTD space or maybe early on in their journey, we're always happy to partner with them to talk about things like where might the clinical trial space be crowded? Where might there be folks impacted by FTD where there's less availability of clinical trials that these companies might want to pursue? And finally, and I should say this is my own wish list, not necessarily representative of other patient advocacy groups or even of AFTD, but if I'm speaking directly to industry, what is our advocacy wish list? And one, Dr. Kolb spoke at length about biomarkers. We hope that FTD can get to the point where we've got robust, valid biomarkers that can help reduce the time to diagnosis. We really need early and accurate diagnoses, and also help a more diverse array of people get an accurate diagnosis, which right now relies to a great extent on clinician judgment. We also look to industry to find different points across the entire pipeline to engage with people diagnosed with patient advocacy groups. And to quote the FDA, engage early and often. For folks that are just thinking about engaging in FTD or looking at maybe whether their asset could be redeployed in FTD, we'd encourage you to reach out to us to discuss that. We encourage a multitude of methods. There's so many different ways to understand the lived experience of different neurodegenerative diseases, whether that's a survey, a focus group, coming and speaking at our conferences. We'd encourage that multitude of arrays. And likewise, the breadth of what patient advocacy groups can offer. Our group, for example, offers funding for early stage clinical trials, all the way to sponsorship to get branding out in front of our community. And finally, we'd encourage you to see the true value. You're not just checking a box because the FDA or other regulatory bodies are saying, hey, we want patient experience data. We think there is real business, ethical, practical value in engaging with patient advocacy and with people with lived experience. Thank you so much for your time, and I believe we have some time now for Q&A.
Thank you so much, Dr. Dodge. And I think before we start with the discussion piece, very quickly to highlight, thank you for very strongly and clearly, educating us that when we think about the asks of our patients and caregivers, the asks are multi-dimensional. There is no one dimension of what patients and their caregiver are asking. So we have to be humble enough to understand. You also put a spotlight specifically in neurodegenerative disorder. Listening to different voices, patients and caregiver voices, and put them together is important. Thank you for also highlighting that words matter a lot. And that's something when we connect science to the right language, that is important for the partner. Now, we at Fortrea, we often challenge our customers respectfully and humbly that how much of what we do day to day, what they do day to day, has been informed and motivated by some of the things which you said. For a company like ours, which has been more than 3 decades around and has all the necessary ingredients to deliver evidence which would be required to run a study which would eventually take a product to a market, it's important that we also, on the operational execution side, keep on talking about how to remain patient-focused and centric. But our customers and our partners are equally, and when I say customer partners, not just clients or pharma company who are building the right solutions, but a whole multitude ecosystem, whether these are our site networks, whether these are our partner vendors, remain patient-centric and focused. And we hope that we continue to do so. I will use the opportunity to now open for discussion and question. And while I understand that there's some little time left, that should not stop, I think, the audience from type your question to chat box because we will take them offline. Off the bat, I have a question for you, Dr. Potter. Shana spoke about her wish list, and I think industry has come a little, maybe I'm not being fair, I think they have come a considerable way from what they used to be, getting patients advocacy and patient caregivers input into the industry sponsored program. Where do you think industry needs to still do more in informing decision internally taking industry, which are more patient-informed patients?
So I think the most important opportunities building on what Dr. Dodge presented really have to do with more systematic encounters up front with the staff, frankly, in big companies across different levels of the organization. So this gets a little complex, but just very briefly, you will have what I call scientific champions in a company, but frequently they're rather low down on the totem pole of making decisions, okay? I hate to say that, but that's the truth. Then there are people in marketing. They have a very different perception. And what you need to do is align those two areas because companies are businesses. And this is where having the marketing leadership and the scientific leadership understand that there is an incredible need, opportunity, and a willingness for people who have conditions to make themselves available for the studies that need to be done to develop novel treatments. And I think what Dr. Dodge was laying out, that they're really trying to build this, but expanding that model more. So there's more of those, more what I call integrated, coherent presentations within the companies involving the major different layers of decision making. Okay.
Thank you so much. I couldn't agree more. And many of us in the room, virtual, have been on the other side of the table, which means have had the pleasure of sponsoring and driving clinical development programs. And I couldn't agree more what Dr. Dodge was saying earlier. It's not just a question of checkbox. It's truly asking our patients where we create value, which would be acknowledged and accepted and utilized by our patients. I think that's an exercise of joint mutual value creation and not just checking a box. I have a follow-up very quick question to you and just a 1-minute question to you, Dr. Kolb, and that is about, you spoke about passionately what it took to finally reach where we are, specifically in Alzheimer's field, to become more precise in identifying what could be the driver of the disease pathology and how do we make intervention. Do you see in future the journey would be this long? You think we can be quicker, will take another 20 years? And what will it take from both patient-centric perspective and how industry respond to it?
I think we will learn from the lessons learned from the AD experience, which in turn learned from the oncology experience, right? And I think the cycle time for doing this in PD or FTD and other diseases will become shorter and shorter, right, as we become smarter on how to do things. And I think the key issue is A, understanding the link between pathology and the clinical symptoms, because you need to understand that to treat it, and then B, to measure it, right, accurately, and then C, to make it accessible to a broad population, right, so that everybody can benefit from it. Those are the 3 components that need to be addressed, right, and we are not even there yet with AD, but I think we will be applying that to other diseases. Already mentioned TDP-43 for FTD, for example, ALS, and alpha-synuclein for PD, and so on.
Thank you so much. Dr. Dodge, I'll come to you. You very kindly presented your wish list, and I think this is the right time to present it, because if I can very simply, think of frontotemporal dementia as a category where you are vis-a-vis the drug development and solution development, I can crudely say that's where the field in Alzheimer's was 20 years ago. And in field of Alzheimer's, over the decades, the patient voice has become stronger, but it didn't start like that. Now, if I can add something or maybe provoke you to say that in your wish list, when you speak about that what industry needs to do, is there something about industry which really irritates you and say, why don't they do this? This is so simple, is easy to do, and doesn't cost much. What would that be so that industry put a spotlight on themselves?
Oh, wow. That's a lot to try to cover in 20 seconds or so. But I would say I would go back to the point of thinking about this early on. And I agree that often there are people in industry thinking about this, and they're not necessarily, at the top of the hierarchy in the companies. But we also we'll hear from companies that, the horse is already out of the gate. They already are under, their clinical trial is underway. It's very hard to pivot at that point. It's hard to pivot their marketing materials. It's hard to pivot, that entire infrastructure. And so I would say, whether you're reaching out to a patient advocacy group or to people that you happen to know in the community that are impacted by the disease, to do it early, to have a conversation. That conversation doesn't cost anything, not asking you to sponsor something or to pay for something or to even hold a focus group, but have an initial conversation with some subject matter experts who really understand the lived experience, not those that understand the biology. I am confident that those conversations are happening, but the people on the ground, the clinicians, the people outside of those clinical trial sites who can really speak to some of those logistics that you need to consider early on.
Really apt word to close the webinar, that is to keep the conversation ongoing, like what we did today. I'll hand it back to Ryan to ensure that we finish in time and make sure that our audience understands how to remain connected to keep the conversation going. Back to you, Ryan.
Yes, well, thank you very much for this very insightful discussion today. I, though, would like to direct the audience to the email addresses that are up on the screen if they'd like to reach out to our speakers for some further discussion. If we couldn't attend to your questions, though, know that the team at Fortrea will follow up with you. I also want to let you know you'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking all of our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank
Thank
you.
you, everyone.
Bye.
Bye-bye.